In this lecture, we will review the diversity of seedless plants. We're now going to take a look at another group of plants, the seedless vascular plants. After plants moved onto land, for the first 100 million years, bryophytes were considered to be the predominant form of vegetation. However, if we look at today's landscape, vascular plants now dominate. The development of vascular tissue was an important adaptation that allowed plants to grow upwards and also allowing them to have better access to light. Seedless vascular plants do not form a clade themselves. Rather, they represent a grade, which is considered to be in organisms that are grouped based on shared biological characteristics rather than being grouped on a shared ancestry. Seedless vascular plants represent two clades, the lycophytes, which are represented by club mosses and their relatives, and monilophytes, which are represented by ferns and their relatives. Like bryophytes, vascular seedless plants also have flagellated sperm, as can be seen in the diagram on this slide. Once released, these sperm need to swim within a liquid medium in order to reach and fertilize the eggs. As a result, these plants are very common in moist habitats. This diagram here represents the hypothesized structure of an early vascular plant, a Gleophyton major. This is considered to be an ancient relative of modern-day plants. Note the branched sporangia as well as the presence of rhizoids to anchor it into the soil. Also note the presence of a fossilized stoma on the branch. We're now going to take a look at the life cycle of vascular plants. Early vascular plants in the fossil record had similar sized gametophytes and sporophytes. However, present-day plants have predominant sporophytes and small ground-hugging gametophytes. We're now going to take a look at a typical seedless vascular plant life cycle. Keep in mind that most of the life cycle will be spent in the sporophyte or 2N stage. We'll start describing this life cycle at the spore dispersal. Once the spores are released from the sporangium, they will germinate and form young gametophytes. These young gametophytes will mature. Upon maturation, they will have rhizoids that will help anchor them into the soil. Remember that gametophytes are going to be very small and anchored towards the ground. A mature gametophyte is considered to be bisexual, meaning that it has the ability to develop an antheridium for the production of flagellated sperm, or it can develop archegonium that will lead to the development of an egg. Once the sperm is released in a liquid medium, it can travel to the archegonium, where it will then undergo fertilization to form a zygote. The zygote will continue to develop in the archegonium. This will then lead to the production of a sporophyte. As this sporophyte matures, it will then produce sporangium. The sporangium will then undergo meiosis to release more spores. Keep in mind that many sporangium may be present together and anchored in the structure called a sorus. Many sori can be present on the underside of leaves. Another derived trait of vascular plants is the presence of xylem and phloem as vascular tissue. Xylem is responsible for conducting water and minerals in a one-way pathway. Xylem tissue consists of tubular tracheid cells that die. As they die, they become strengthened or lignified upon maturity. This also helps to support the plant and allow for vertical or upward growth. Phloem, on the other hand, conducts sugar, amino acids, and other organic material in a two-way flow. Phloem tissue consists of cells also arranged into tubes. However, they remain live upon maturity. Take a look at the diagram below to see the difference between xylem and phloem tissue. Another derived trait in vascular plants is the evolution of roots. Sporophytes, because they were independent, developed roots. These are considered to be true organs that absorb water and nutrients from the soil. Roots also serve to anchor plants and provide support for upward growth. In the fossil record, there is evidence that depicts similarities between root tissue and early plant stem tissue. This indicates that roots may have developed from the lowest ground portion of stem tissue in ancient ancestral plants. It's currently unknown if roots evolved once in a common ancestor or independently in multiple lineages. Another derived trait in vascular plants is the presence of leaves. Leaves are the primary organs that carry out the process of photosynthesis. Leaves are typically flattened in order to maximize the surface area that captures solar energy. Leaves can be classified as two major types, microphyls and megaphylls. Microphyls are considered to be small spine-shaped leaves that are supported by a single strand of vascular tissue and are only present in lycophytes. Megaphylls are larger leaves which have an extensive vascular system that extends throughout the leaf. These leaves are present in all vascular plants. Please consult the image below to see the difference between microphylls and megaphylls. Another derived trait of vascular plants is the presence of sporophylls. Sporophylls are considered to be modified leaves that contain sporangia for reproduction. The undersides of these leaves have clusters of sporangia, commonly called sori. These can be seen on the image to the left. Some sporophylls in some plant species may be grouped into cone-like structures that we call strobili. These can be depicted in the image to the right. Most of the seedless vascular plants produce spores that are homosporous. This means that these spores develop into a bisexual gametophyte that can produce both eggs and sperm. Seeded plants and a small amount of vascular seedless plants are considered to be heterosporous. This means that they produce two types of spores, megaspores that develop into female gametophytes or microspores that develop into male gametophytes. The female gametophyte can then only produce eggs while the male gametophyte can only produce sperm. Seedless vascular plants are currently represented by two clades, phylum Lycophyta, which includes club mosses, spike mosses, and quillworts, depicted to the left, and phylum Monilophyta, which includes ferns, horsetails, wisp ferns, and their relatives, depicted to the right. Ancestral seedless vascular plant species dominated the first forests that were present during the Devonian and early Carboniferous period. These seedless vascular plants were responsible for causing a distinct drop in atmospheric carbon. This eventually led to a global cooling event. It is believed that the reduction in carbon levels was due to two reasons. The first reason being that the roots of these plants secreted acids that released calcium and magnesium from rocks. These reacted with the carbon dioxide in rainwater to form carbonate compounds. These compounds ran off into the surrounding oceans, ultimately trapping their carbon in marine-based rocks. Another reason for the reduction in carbon levels was due to the fact that the plants did not completely decay. This is because they were present in a semi-aquatic environment. These plants rather formed layers of peat in the stagnant waters that were present during the Carboniferous period. Eventually, these ended up forming coal over time as the heat and pressure increased over millions of years, trapping the carbon in the coal particles. Nowadays, burning coal will release this ancient carbon back into the atmosphere and now is a major contributor to global warming.